In this part of our video seminar series, I'll turn to the evolutionary questions addressed by our work on mitochondrial temperature. As almost everything else in the second half of this series, there is much speculation and little hard fact to draw upon. But whereas the questions we have considered already and shall consider in the future that relate to cell biology and pathophysiology are susceptible to experimental test, when we come to evolution, it's almost impossible to conduct real experimental simulations, and we must instead rely mostly on indirect evidence and the historical record. So take this as a word of warning, applying even to the most ingenious or seductive evolutionary hypotheses. Last time, we considered the potential implications of elevated mitochondrial temperature for a number of cell biological phenomena, including mitochondrial dynamics, apoptosis, immunity, and heat export by thermogenic cells. In this segment, we'll look at some major evolutionary issues on which there is no universal consensus, but nevertheless an increasing amount of data pointing tentatively in specific directions. We'll start by considering whether aerobic bacteria, the ancestral precursors of mitochondria, might also be hot. This will take us more specifically to the origin of eukaryotic cells, and then on to the emergence of homeotherms. Organisms like ourselves maintain a more or less constant internal temperature, all within a few degrees of 40 degrees Celsius. Try any AI-powered search engine, and it will tell you emphatically that bacteria are not homeotherms and simply exist at the temperature of their environment. Yet nobody has, to my knowledge, actually tested this. A moment's thought should alert us to the fact that aerobic bacteria have a very similar energy transduction system as mitochondria, since they are evolutionary relatives. The efficiency of this system is such that much heat should be released just as in mitochondria. Although eventually dissipated to the surrounding medium, it's reasonable to assume that the heat remains partially trapped in the cell itself, as it is in mitochondria, raising its temperature. In fact, the OXPHOS system of bacteria, though composed of the same core proteins, is rather simpler than that of mitochondria, in that it lacks many of the peripheral or supernumerary subunits found in the mammalian, plant, or protist and OXPHOS complexes. It seems either to have become stripped down to its essentials, or else these extra subunits were never present ancestrally at all, and there remains much debate and speculation over their precise functions in mitochondria, to which I'll return in part 8 of this series. One possibility is that they provide an element of thermal insulation, such that in bacteria, the heat generated is more readily transmitted beyond the OXPHOS membrane, and the internal cellular temperature is therefore actually less than in mitochondria. Importantly, the architecture of bacterial cells is rather similar to that of mitochondria, at least in gram-negatives, which have a double membrane, although they also have an external cell wall. Gram-positives have a very thick cell wall structure in addition. These multi-layered sheaths have traditionally been thought of as providing defense against environmental toxins, predators, and bacteriophages, as well as enabling various signaling systems and preventing leakage of cell components. But they must also provide a layer of thermal insulation, perhaps even more effective than that enjoyed by mitochondria. For an organism like E. coli living in the gut of mammals at 37 degrees Celsius, it's reasonable to assume that their internal temperature is much hotter, more like that of mitochondria. But, as stated already, nobody has yet actually measured it. People still have a hard time grasping that this is a serious possibility, so I hope it will soon be tested by someone, and the concept either validated or laid to rest. One bacterial species group that has attracted great attention is Wolbachia, intracellular symbionts found in a great many arthropods, including Drosophila. When resident, they have diverse biological effects, notably on reproduction and behavior, but also on immunity. they are also phylogenetically close relatives of the ancestor of mitochondria. And by the above logic, combined with our observations on mitochondrial temperature, should also exist within the cell, within the host cell, at elevated temperatures. It will therefore be of great interest to measure their temperature using tools such as I described in part 2 of this series, notably the ratiometric fluorescence of the BG-temp proteins. Does their heat output affect the temperature of their host cells? How does it respond to environmental fluctuations? And could heat production underlie some of their biological effects? Before turning to the link between temperature and the emergence of eukaryotes, it's appropriate to review very briefly the currently held view on the nature of the classes of cellular life forms on our planet today, and where they came from. Whereas 60 years ago only 2 such broad classes were recognized, prokaryotes, small single-celled organisms lacking a true nucleus and with a minimal internal cell architecture, versus eukaryotes, whose cells are typically at least 10 times larger, with a separate nucleus containing all or almost all of the cell's DNA, plus a complex endomembrane system and organelles dedicated to specific metabolic tasks. Gradually, however, this view has been modified in 2 fundamental ways. First, by the discovery that prokaryotes are really 2 classes of organisms as diverged in time and genetic distance as either is from eukaryotes, arguably more so. Second, by the understanding that eukaryotes are really descended from a cellular mosaic of particular lineages of archaea and bacteria, with an archaeal relative of the so-called Asgard class playing host to a bacterial endosymbiont related to present-day alpha proteobacteria, which then gradually evolved into present-day mitochondria, and later on to a member of a separate group of bacteria, the cyanobacteria, that evolved into the photosynthetic chloroplasts of plants and algae. A full understanding of the evidence that led to these insights is obviously beyond the scope of these videos, but it's important to grasp the basic idea of endosymbiosis before we proceed. The generally accepted idea has been that by joining forces, the archaeon was able to make use of the much more efficient metabolic route to generate ATP, i.e. OXPHOS, that had evolved in aerobic bacteria, while the bacteria were able to plunder a rich and concentrated source of substrate. Informed by our findings of elevated mitochondrial temperature, my colleague Corey Dunn has put forward a radically different idea to explain the evolutionary origin of eukaryotes. There is a generally accepted view that archaea, probably prokaryotes in general, originated in the vicinity of oceanic hot springs, hydrothermal vents, where the biochemical reactions of life were first energized by heat emerging from deep inside the earth under anaerobic conditions. Dunn's idea is that bacteria gradually evolved in a different direction, able to thrive in a cooler environment by using the oxygen released by photosynthesizers to release the energy locked up in organic substrates, yielding ATP much more efficiently, but releasing heat in the process, so essentially keeping themselves warm. By these 2 classes of organisms forming a partnership, the aerobic bacteria were indeed able to source substrates much more easily than from the dilute ocean, while the thermophilic archaea acquired an alternative source of heat, so could themselves colonize cooler environments, the oxygen-rich ones where the aerobic bacteria were most able to function. In time, the partners became inseparable, with the bacterial heaters taking up residence inside the cells that were the source of their substrate materials, thus maximizing both substrate supply and heat acquisition. This hypothesis has received strong indirect support recently from the inference that the ancient archaeal host for endosymbiosis was a moderate thermophile, based on deducing the ancestral sequence of a key component of the protein synthetic apparatus and measuring its temperature optimum experimentally. I now want to turn to a more recent evolutionary step that I believe must also involve mitochondrial heat production, namely the fact that we've already alluded to, that all mammals and birds today have body temperatures close to 40 degrees Celsius, despite their hugely variable sizes, lifestyles, habitats, body architecture, and sources of nutrients. This has always puzzled me, and the explanations advanced by others, there is something magical about 37 degrees for optimal protein function, seems implausible to me, given that so many organisms thrive perfectly well at environmental temperatures far from this, from below zero in the polar ice sheets to over 100 degrees in hot springs. Other organisms, especially for example some lizards, can accommodate environmental temperature fluctuations of 50 degrees or more. Instead, I'd like to offer the following hypothesis. There's a broad consensus that vertebrates first colonized the land about 280 million years ago, and that ocean temperatures at that time were around 40 degrees, probably a little lower. Marine vertebrates at that time naturally had a body temperature of this order, with mitochondrial heat probably keeping them just a little warmer than the surrounding ocean. Exposed to the air, their rate of heat loss would be much greater, but all of their proteins would have evolved over a long period of prior time to function optimally in the 40-degree range. Of course, over evolutionary time, all 20,000 or so protein-coding genes could have evolved slowly so that the proteins would function at lower steady-state temperature. But a one-step solution would be far more likely to have survived and thrived in this onshore environment. That one step for me was obviously an increase in mitochondrial heat production, either by having more mitochondria per cell or by increasing their heat output by a decrease in the thermodynamic efficiency of OXPHOS. The former would be limited by the ATP needs of the organism, though burning off the excess ATP would equate to the latter. The evolution of insulating systems such as fat, feathers and fur obviously helped. Again, essentially a one-step process. This concept already shadows ideas first put forward over 20 years ago by Doug Wallace on the evolution of human mitochondrial DNA that is discussed in the next part of the series. The corollary of the main hypothesis is that organisms, including vertebrates and other chordates, that did not shift to permanently living on land, but instead remained in the oceans, have body temperatures more reflective of their current oceanic environment, since ocean cooling over the intervening 300 or so million years has been very slow, enabling the rate of protein evolution to keep pace with it. Today's aquatic mammals and birds that moved rather abruptly back to the sea when its temperature was far below that when their ancestors colonized the land, have continued to evolve body adaptations to insulate themselves and thus retain body temperatures in the 40-degree range. It's now time to preview the next video in this series, when we'll look at some of the implications of hot mitochondria for physiology and disease, starting with cold resistance and obesity, then looking more closely at classic mitochondrial diseases, and finally the pathophysiology of the heart. How does mitochondrial heat production influence life in a cold climate, for example? And could mitochondrial genotype predispose to metabolic diseases in people who abandon traditional diets? I'll close here by acknowledging my funders, sponsors and colleagues, especially Corey Dunn, who has proposed the heat hypothesis to explain the origin of eukaryotes by endosymbiosis, as well as members of my own team who helped develop some of the ideas outlined here. I also gratefully acknowledge the sources for external images I have used in this segment of the series.